I think this is third event I attend. The third time I attend the event related to K-computer. The first time was in Osaka University. The second time was here. This is the third time. So I think this is something special. I've been developing very good relationships and friendships with my Japanese colleagues, and thanks for having me again. Let me explain my title. This title is a bit complicated, mouthful. So there are three things here. So there's the molecular dynamics simulation. That's one thing. Then there's the structural biology. That's another thing. And the third thing is called large biomolecular assemblies. So that's three things. I try to connect these three things. And the word is emergent, meaning it's not quite there, but it's becoming something that you can start to see the potential. Instead of just being sort of a supplementary tool of structural biology, it's my hope, or at least my effort in my work, to make it to play a more central role. So that's basically what I'm trying to the message I try to bring. Okay, so here's the molecular dynamics simulation for those people, right? Everything is a particle. Even the red background, they are water molecules. They are so fine particles. Everything's particle, and the particles are following the Newton's law. And they bump into each other, and they move, right? Thermal motion. So everybody have to cite this Richard Feynman quote saying, everything can be understood in terms of jiggling, wiggling of atoms. That's not true. That's not true, right? You don't simulate the atmosphere by simulating individual atoms, right? And that's not necessarily true. But for now, we don't seem to have a better way, and I think in the future, a better way might emerge. Not everything has to be understood in such a reductionist way. Understood the particles moving around. That's just not how you simulate the atmosphere or the climate. Okay, so now just a little introduction of molecular dynamics. This is, you start with the DNA, then DNA information was transcribed to RNA, RNA information was transcribed to protein. And the protein is a polymer, with 20 amino acids, and that's a polymer. And then this polymer folds into a relatively well-defined shape, right? But it's a relatively well-defined structure, but it also has dynamics. It can move. So it's a little micro machines. It's sort of it has a well-defined structure and a well-defined dynamics. So that's what is complicated. So if you look at the one amino acid, here in this case it's a tyrosine. It looks like this. You have this 6-atom ring, and so on and so forth. And this is how a typical small protein would look like. And then these atoms are connected by chemical bonds, and these chemical bonds also have a thing called the dihedral angle controls the rotations of these entities. So right, and so you have this the rule of physics to control these motions, and ultimately it's I'm not going to the details. There's the Coulomb's law because charges interact, and I'm stealing slides from Michael Levitt's Nobel talk. So I have to acknowledge that. But you have these energy potentials that can be translated into forces, and from forces, by Newton's law, you can translate into motion, right? So that's just molecular dynamics. And a brief history of MD, molecular dynamics. So the first piece is basically they invented the Monte Carlo method, right? So this paper has, I just checked, it has like 35,000 citations. And people don't even cite it anymore because, it's just a part of our legacy. It simulates a liquid system of 2D spheres bumping around with each other, right? So 1953, 55 Fermi, Ulam. Ulam is the guy come up with the hydrogen bomb idea, the implosion. Pasta must be a big guy, but I'm less familiar with. So it's a 1D crystal, and 1956, first proper MD simulation. By proper MD simulations, I mean it's not Monte Carlo, it follows Newton's law. Then 64, MD simulation of argon liquid. And 71, a simulation of water, MD simulation of water. So it started with at 1953. Why? Why? Because computer. Because that's the time computer becomes a real thing and people start thinking about how to use it. The idea itself, in terms of physics, is not that hard, but it's crazy to think that you can do, manual calculations by this, in this approach, right? When a computer comes along, so MD is intrinsically connected to computer. And 1973, Kopper's group published the first MD simulation of proteins in picosecond time scale. 80s and 90s, a lot of methodology development. 98, for small molecule, microsecond time scales reached. But it's a very small protein called the villain headpiece. I will show a movie later. And 2009, Anton supercomputer came along. It's specialized for MD, and that brings the simulation time scale to micro to milliseconds. Sorry. So 2013, Koppas and Washaw and Levitt won Nobel Prize for MD simulations. So, so a brief history. So like I said, the crucial thing here is time scale, because if it's a movie, you don't take your girlfriend to see a movie for a 1 second movie, right? But simulation for a long time is like a 1 second movie. Like it doesn't tell the story because the story takes 2 hours to tell, and the snapshot or 1 second thing doesn't tell the whole story. So how do you reach that time scale? In this time we used a very dumb method, right? Very straightforward and engineering method. We put the best Silicon Valley engineers together, and we built a super specialized computer with the goal of reaching the 2-hour time scale to tell a good story. And the 2-hour time scale for movie translate to millisecond time scale for our simulations. So, right, and in the movie, I will labor this metaphor. In a movie, the basic unit is one snapshot, right? Then you run many snapshots to make a motion picture, right? And in our MD simulations, you have the same thing. That's in the femtosecond time scale. That's your minimal unit, and you have to start with femtosecond, and to reach milliseconds. So you need 1 trillion steps to that's a challenge, right? Okay, but then I hope there's music or something, then there's Anton, sunshine comes out, but it really was a straightforward but very effective idea to advance this field. I was very fortunate in the sense that I was out of job in the right time and bump into a professor from Columbia University who happens to know me and who said, "Oh, would you be interested in getting a job in New York?" And I was like, "Sure. What is that?" And he said, "I don't know, but the guy's has a lot of resource, and the guy's very dedicated to science, and the guy's nice, and he wants smart guys to join him." And I thought, okay, it's New York, it's good. And by the way, the alternative would be that I'm out of job, I might be deported. So I think that's an easy decision to make. So I joined them very early on, with a handful of people at the beginning for the first year, and it was a lot of fun. I must say it's a lot of fun. So the earlier work on mine, I guess the more, the published one was this Gaussian split Ewald GSE method to solve the Poisson equation, which is tailored for this kind of chip design. I did talk to the engineers and understood what is easy for them, what is hard for them, and at the end we come up with a new mathematical formulation of the algorithm of the problem, and that comes with the algorithm, and that was incorporate the algorithm was incorporated or hardwired into the chip. So that was very gratifying. I guess I also worked on a lot of boring things like figuring out how many bits we need to do accurate enough electrostatic calculation. The number is 23. You need 23 bits for the fixed point data storage, so on and so forth. So I did a lot of random things with my colleagues, and then the professional chip designers came along, and they took over the job, and they did a fantastic job in designing the chip. And the chip was the first generation Anton chip was manufactured in Fujitsu, and this is 2008. That's the day we got the chip back. We can touch the chip. We can put our fingers on the chip, and so we had a little party there, and I think I use this picture because it make me look more important to the project than it really was, but and so this is the module for electrostatic interactions. So these are the algorithm, we worked out and put on the chip. So this chip can do nothing. This chip can do nothing other than MD. You cannot run this PPT. You cannot run this PowerPoint. You cannot do a simple calculation. It can only run MD simulations. And Steve talked about NVIDIA and GPU, and a lot of ideas sound very familiar to me. And I think there's basically if you have to put it simply, the idea behind Anton is to stuff the pipeline and keep it going and keep it streamlined. So I'm not I'm not qualified to really get into the details, but this is Anton and the MD simulations. Now let's talk about structural biology. Now the first half of 20th century in terms of science, I think is about physics. It's about quantum physics. It's about relativity, and it's especially about quantum physics. I think the second half of 20th century in terms of science, I would venture to say that it's about the structural biology. So 1951, Linus Pauling come up with this beautiful idea of a helix. He said, okay, this helix looks really cool. And I think it's too cool to not be true. And in proteins, you must have helices. And he's right. He's right. And this is the helix. Right? And 1953, again, I'm stealing Michael Levitt's Nobel talk. Crick and Watson. I don't know why Watson's picture is not here, but actually the order is Watson and Crick in the publication. But people seem to have some bias. But they come up with the double helix idea, the DNA model of double helix, and that was like a monumental achievement. 1959, the first protein structure, detailed protein structure from X-ray crystallography. Right, that's a myoglobin, and so on and so forth. In 1963, you have hemoglobin, and now protein structures from X-ray crystallography and from cryo-EM is being deposited in the number of tens of thousands every year into the database. So that's routine. It's growing exponentially. And the name of the game in that field now is about these large. This is a mistake. There's nothing confidential about this. It's about these large assemblies of biomolecules. In this case, this thing is called a spliceosome. It's hundreds of proteins come together, form a machine to manipulate RNA, to cut a piece of RNA out. This is the machinery. What this machinery purpose is for, and this is just to give you an example. This is 2015's work. It's about large biomolecular assembly. That's my title, large biomolecular assembly. So, but obviously this is from experimental methods, from cryo-EM. Cryo means very cold. E means electronic. M means microscope, right? So this is a structural biology. I want to make a point that the first two most important structural biology work were modeling work. They were not directly from experimental data. They utilized experimental data, but they were from Linus Pauling's head. They were from Crick and Watson's head. So they were modeling work. They didn't use much computers, but they were doing modeling. And I think we are following that great tradition. And later, the structural biology became dictated by experimental work, X-ray crystallography, NMR, and the cryo-EM. But I think at a certain point, we want to revisit that great tradition of structural biology with the help, with an enabling by supercomputing. So that's basically what I'm trying to do, and trying to. I'm part of that effort.